Yeah, first of all, thanks to the organizers to really do this workshop 1 year after it was planned. And I think it was very nice to have this kind of introduction of everybody. It was very nice to see yeah, so many interesting topics covered. Yeah, I will talk about Microswimmer Reinforcement Learning with genetic algorithms. And it somehow covers two topics. On the one side, Low-Reynolds-Number microswimming, and on the other hand, machine learning. And this work has been done at the TU Wien, together with Benedikt Hartl, Maximilian Hübel, and Gerhard Kahl. But now I'm a postdoc with Christos Likos at Uni Wien. And as I said, it will be also a little bit about machine learning, but I think somehow different than it is commonly used, in the sense that we do not use machine learning to solve an outstanding problem somehow or an efficiency problem, but to use artificial neural networks to understand kind of internal decision-making machineries, here applied to microswimmers. So a quick outlook. First, I will quickly say something about microswimmer chemotaxis. This is somehow our model system in 1D. And then I will show you how a 3-sphere swimmer, which is a very simple model microswimmer we use, is able to learn swimming with genetic algorithms applied to artificial neural networks. Then I will come how such a microswimmer in 1D can perform chemotaxis, in the sense that it can sense gradients motivated by biology, essentially, because normally a microorganism cannot directly detect gradients, but either use some sort of spatial sensing or temporal sensing, and I will talk about this a little bit. And finally, I come to the fact when a neural network or an internal decision-making machinery is not able to detect signals perfectly, but there is noise involved, then we see that in our procedure, the well-known run-and-tumble motion yeah, pops up. Okay, so here's just one slide about microswimmers. I guess in this workshop, we will hear more about different kinds of microswimmers. In principle, a microswimmer is just a microscopic microscopic object which can move by its own without external forces in a liquid. And there are in principle two big classes: biological microswimmers, like microorganisms, or artificial microswimmers. I guess you will hear about both types of swimmers a lot during this workshop. There are actually, what I think, there are two main differences between biological and artificial microswimmers. One difference is somehow the locomotion strategies. So the artificial ones have typically different strategies. And also what is important is that biological microswimmers usually have an internal sensing machinery where they can make decisions and decide where to go, where to swim. Whereas for artificial microswimmers, there is more external control. So what I'm talking about today is decision-making of microswimmers in the context of chemotaxis. I just show you here 3 examples of organisms, biological organisms, which can do chemotaxis. For example, the eukaryotic E. coli bacterium, prokaryotic, or the eukaryotic cells, or also multicellular organisms like worms, elegans. How they sense the environment and react to the environment is different. For example, E. coli bacterium, they have some sensors in the membrane. Then they have some internal machinery with some feedback loops, where eventually there is some decision about if the flagella motor attached to the cell body should go to the clockwise or anti-clockwise direction. Yeah, in this sense, the input through the signaling, the sensing is coupled through the signaling towards the output, let's say, of the swimmer. Eukaryotic cell, and I have to say these E. coli bacteria typically perform temporal chemotaxis, which means that they can not sense spatial gradients of chemicals, but only temporal gradients. That's different from many eukaryotic cells, which can perform chemotaxis. So they also have receptors, and then there is a whole bunch of internal signaling going on, which essentially drives internal processes which influence the locomotion of the cells, for example, through actin polymerization, yeah, which then helps cells to polarize and to do chemotaxis. Another class, so and I have to say, these single cell organisms, they do it with some signal network within the cell. But the larger organisms like C. elegans worm, it's a multicellular organism consisting of, and it has around 200 or 300 neurons, and it can use some sensory neurons coupled to a motor system, to motor neurons, to make decisions about the movements. Yeah. And what is important to notice that there has a lot of work been done on decision-making machineries of organisms. But we want to study, or what the challenge is, let's say, is to study a system, a very simple system, where this decision-making machinery is coupled to a physical environment. So because if you make a decision, like if you want to go left or right, you have to couple it to the environment, so to the physics. Yeah, you cannot just decide to go to the left or to the right. You really have to couple it to your motor system. Yeah, and this depends on the environment. And what I will talk about today is a viscous environment where microorganisms can swim in it. Yeah, this is somehow represented through this linear relation between velocities and angular velocities and forces and torques. Yeah. Okay, this is somehow the setting. And okay, so I have to speed up a little bit, but I thought it's good to somehow give a motivation also about this. What we use is a model microswimmer, a 3-bead swimmer. So I guess many of you know this. It's essentially a swimmer which consists of 3 beads, where forces can be applied in one dimension, and it moves force-free through a viscous fluid. The arm velocities are just the change of the arm length over time. And there exists, for example, if you use far-field hydrodynamic interaction between the moving beads, a linear relation between the applied forces and the velocities of these beads. So that's a well-known problem for a microswimmer. And what is very common in literature is that you prescribe an arm motion of this, of the arms. For example, you have this rectangle here, moving at certain velocities. These are these 4 points here in the velocity space. And then you can calculate the forces, so these 4 lines, the forces you need to get this motion. Yeah. What we have in mind is a different approach. We want to find a policy for the forces and then calculate the arm velocities and the mean swimming speed and so on, and the arm length. And to this end, we will limit the forces on the beads to a certain maximum force, and also the arm extensions by applying a harmonic restoring force. So if the lengths are more than 30% larger or smaller than an equilibrium length L0, which is 10 times the radius of the beads. And then we get typical force curves, velocity curves, and curves in the arm lengths of the microswimmer. And what we now want to do is to find an optimal strategy how to solve this problem when the forces are limited. And for this, we use reinforcement learning with artificial neural networks, so how is it done? In principle, you have an environment, let's say a viscous environment, and then you have a state, which is for example, given by the length and the velocities of the arms, but also by the chemical environment. I will come to this. And then the microswimmer proposes an action, so an output on the forces F1 and F3. F2 essentially, due to the force-free condition, can be inferred from the forces F1 and F3. And in such a way that some reward is maximized. And the reward can be, for example, the distance a microswimmer swims in a viscous environment, or also to swim towards chemical sources. So, and you typically have these reinforcement learning cycles where you want this reward to be maximized over time and where it eventually leads to a plateau. So that's the idea. So here's just a small example. When you have the input vector x, for example, given by the state of the swimmer, the lengths of the arms L1 and L2, the total length LT, then the individual arm velocities V1, V2, and the total arm length VT. And then the thing is, very often when artificial neural networks are applied, you can have very complicated deep neural networks. We found that we don't need very complicated neural networks, but rather simple ones. But some machinery, sensing machinery between input and output is in principle given by a hidden layer or some hidden neurons, which represent this, let's say, some sensing machinery. But in the case where you have only direct connections, for example, between input and output, you can simply write the output of the forces as a nonlinear function of the input x. This is essentially what neural networks are doing. It's just a nonlinear operation where you take an input and you add a weight. And here, sorry, that's the weight and that's the bias. And then you apply this nonlinear function, which in our case is a hyperbolic tangent functions, which automatically assures that the forces are between minus and plus F0. Okay, so quickly, how do we do the training? We use the NEAT genetic algorithm, and it can do reinforcement learning. NEAT means it's a neuroevolution of augmenting topologies. So it uses genetic methods, and it optimizes the structure of the artificial neural networks, the topology and the weights to maximize the reward, and also removes unnecessary links between nodes. It uses an ensemble of artificial neural networks, for example, an ensemble size of 200. And you can calculate the fitness for all the networks. And the ANN solutions with the large fitness are retained and preferentially selected for reproduction to form a next generation of INNs. And then the good traits of the controlling networks will prevail over time, and it will direct the entire ensemble to the desired solution. And it's doing this with these neuroevolution techniques, like using mutations and genomes and genotypes and networks phenotypes. Okay.
Hi, sorry, can I stop you for a question, or shall I ask it later?
Maybe later, because I'm already a bit late. Yeah, if it's okay. Cool. Or is it a quick question?
Yeah, I missed what's the reward.
Sorry. The reward, the reward can be, for example, the distance a swimmer moves in a certain direction. So it depends actually on the specific task you want to do. So the first step, and that's exactly what I want to tell you in a moment, is when you just want to learn unidirectional motion without any chemical field, then the reward is just to maximize, for example, the plus x direction of the center of mass distance of the swimmer in a certain simulation time ti. That's the reward. And then we also define a fitness, which is simply the mean swimming speed. And this is just the reward per time. Yeah, this is just the swimming speed. And then, so here you can see on the left-hand side a typical evolution of a neural network, and the reward over time here. So the network very quickly goes to kind of an optimum policy. And how does this look like? Actually, the optimum policy is that you get this square-like shape in the force space, whereas the resulting dynamics of the arms is not a rectangle, yeah, like it is often done in simple theoretical models. And then you see how the network evolves. What is interesting to see is that the NEAT algorithm tries to maximize the reward, but it goes to a quasi-optimum state very fast, and then it's just trying to add a little bit more links or hidden neurons, and then you get maybe 1, 2, or 3% more reward, but not more. Yeah. You can also redo the training runs. So here are 10 training runs, and it always goes here to this kind of maximum reward. What is interesting to see is that the NEAT genetic algorithm finds different strategies. So neural networks of different complexity. And here you see the complexity shown versus the fitness. So the maximum fitness we find, so that's the optimum policy, is around 1.35 in these units. And what is interesting is, so the complexity is here measured in terms of number of connections in the network and number of hidden neurons, that there are already very good solutions by only using 2 connections between output and input neurons without using any hidden neurons. Yeah? And also, if you go to this kind of optimum fitness solution branch, you only need 4 connections to have an optimum policy. So essentially, a microswimmer can learn to swim in an optimal way with exploiting very few number of connections between output and input neurons. Yeah. So here you can see typical trajectories for different networks shown here. And here is the phase-space dynamics. I just want to move on a little bit. But before that, before I go to the chemotaxis part, I want to quickly show you how this policy looks like dynamically. So here you have, in the simple case where you have only 2 connections between length 1 and force 3 and length 2 and force 1, you can see here how this is essentially the hyperbolic tangent function. And this ensures essentially not optimal policy, but this very good policy for this microswimmer in a very simple way. I want to skip this. I just want to say you can map now, if you substitute the forces by the lengths of the swimmer through to the learned policy, you can map the system of the 3-bit swimmer to a dynamical system where essentially the swimming versus non-swimming solution is a bifurcation here in this 2D phase space. Yeah. Okay, but now I want to also show you that we can learn chemotaxis, and here the reward is modified. Yeah, when a microswimmer moves in a chemical field, the reward is now to move in a certain direction in a, where D, so and D is it's the direction of the chemical field where it is. Yeah. So if it goes in a wrong direction of the chemical field, so if it goes away from a maximum, it's a negative reward. Yeah. And the thing is that the microswimmer does not per se know what the sign is. So it doesn't know the gradient directly, but it has to apply, it can only do temporal or spatial sensing. And to this end, we extended our approach. So we have the state of the swimmer, but now also the chemical field, which couples or which goes to a so-called chemotaxis agent, which approximates the sign D in which direction the microswimmer wants to go. And then this is applied to the learned network I showed you before, and also modulated by a so-called permutation layer. Yeah, it wants so there is a possibility it goes to the left or to the right direction. Yeah. And so, for example, for the spatial sensing setup, we say, okay, the swimmer cannot measure the gradient, but it can measure the chemical field at the 3 bead positions. And then the NEAT algorithm has to find a policy to estimate the gradient D via this variable G. I don't want to show you how how the training was done here. I just want to say that the solution looks very simple and very sensible, that this 3-bit swimmer just uses the chemical field at the outer bead position and the inner bead position with weights which are similar for these two beads, and it connects it to get to approximate the gradient. Yeah, just by saying, okay, just use the sign of the difference between these two chemical fields. It's very sensible. The temporal sensing is essentially a little bit more involved. So we have somehow include some recurrent mechanism there. Because of the temporal sensing, we included a so-called chemical memory control cell. I don't have time to go into details. I just want to say that this there is an internal memory which is coupled to the chemical field at the center of mass position. This is the CC. And then the neat algorithm finds a simple neural network to perform this temporal sensing. Yeah, I just want to show you here the evolution of this neural network. Here we have chosen a different approach, a more stochastic optimization, in order to explore a broader range of networks. Yeah. And essentially here for the temporal sensing, you need some hidden neurons, about 5 hidden neurons in principle, and you can do very good chemotaxis. Here are some trajectories, but I want to show you maybe a movie. This is just an outcome. Here the network is not shown dynamically, but just how the swimmer is doing. It tries to go to the maximum of a Gaussian field, and then it has to decide if it wants to go to the right or to the left. This is somehow represented by these two colors in the phase space. Yeah, this is then regulated by this permutation control layer. And then it learns to somehow to switch between moving to the left and to the right a little bit, and is doing these oscillations around this maximum. While what is interesting, the spatial sensing swimmer is not doing these oscillations, but it stops really at the maximum. Yeah. Okay.
All right.
Okay. So I will maybe stop here, and yeah, thank you for your attention. If you have questions, please ask.
Okay, thanks, Andreas. Do we do the questions just by audio? I mean, let's maybe try that, right? So if you have a question, just go ahead for now, or put it into the chat that you want to have one later.
I do have one, if I can.
Okay, go ahead.
Hi. Thank you very much. It was very nice. And when you talked about the chemotaxis problem, it reminded me quite a lot about these bandit problems that people use in reinforcement learnings. Do you know if there is any formulation of this problem as a bandit kind of problem, if you know about bandit problems?
No, sorry, I'm not aware of what you're talking. Which kind of problem?
It's like bandit. So the agent tries a move and then he sees a reward, which in this case it will be, you know, you try to move somewhere and then you see if the chemical has increased or not.
Yes. Yeah. Yeah, as I said, I mean, we just took these two strategies motivated by biological systems like spatial and temporal sensing, and yeah.
I saw a raised hand here, but now I cannot lost the person in the gallery view. So it was Pier Francesco or something like that. I'm really sorry for that.
Yes. Hi. Just a brief question. Do you have a cost on the power the swimmer can use, and how the results that you get change with the cost?
Yeah, no, that's a very good question. So in the moment, we only have one constraint, that the total force on the beads is constrained, but there is no kind of cost in terms of, for example, you could constrict the total energy in your system and so on. So these are things we're thinking about to apply, but yeah.
And I have another additional question, if I can. So you have like, I think 3 or maybe 6 variables you are trying to optimize. Did you try to do it just with optimal control without going through reinforcement learning?
So actually, I also, no, so optimal control, no, I didn't, we didn't do that. I suppose that the solution should be the same in terms of phase-space dynamics, but yeah, we haven't done this yet. We have only solved it with the neural networks. For the 3-bit swimmer, it's not a very complicated system, but you're right. I mean, one should maybe also compare it to this kind of optimization methods. Yeah.
Thank you.
So maybe I take the chance now to ask a question, maybe a very naive one at first. So this is purely one-dimensional, right? So the swimmer cannot change its orientation, just go to the left or right. Yes,
exactly. So here I just show a video where we extend now our system, our approach to 2D or 3D. For example, we have this kind of breaststroke-like swimmer, which learns to do these breaststrokes and to do translation and also rotations. So we are now trying to apply this to more kind of realistic microswimmer settings. Yeah, but the things I talked today was just in 1D. Yeah.
But for the one-dimensional case, can you also do an analytical solution of this optimization problem?
Yeah, no, actually you can't. So we also tried, but it's not possible. Yeah, so. Yeah.
Okay.
At least I don't know it, so we tried, but no.
Okay, we have another question from Gaurav Prakash Srivastav. He says, thanks Andreas for the nice talk. Is there a maximum on the reward?
Yeah, so actually our policy learns the maximized reward somehow. So there is an optimum policy which gives you the optimum reward. If you like. Yeah.